Hello and welcome to Sarat Chandra IAS Academy, where your dreams are a mission. This is Yatharth here. Today we'll be talking about the GE mustard. Please take care, it's not GM mustard. And what is it? We'll see. And how it differs from GM mustard. Alright, so this is a way forward solution you can write in your JS3, Science and Tech and Agriculture. And let us see how they differ and what is the difference. In the background, you can see a picture of DNA. So we'll see how it correlates to DNA also. Alright, so first of all, let us see what is GM. Okay, GM basically means genetically modified. Okay, modified means having any kind of changes in the genetic data. So as we know, and many of you may also not be knowing, that the basic building blocks of everything, every structure that is on earth, if it's non-human made, if it's natural, are basically proteins. Of every living thing is protein. And protein come as an expression of DNA or RNA. The basic function of DNA or RNA is to make proteins further on, or to give instructions that how and what proteins are to be produced. Okay, this is the basic fact of life. This is the basic work of any nuclear material or genetic material. It tells the cell how to make protein or what protein to make. Alright, now how it tells? Then there must be coding, which is decoded by the cell. Okay, so DNA converts into RNA. RNA tells the code. Alright, DNA is just a more stable form, double helical form of the single straight RNA. So now when we talk about the coding in the DNA or RNA as a genetic material, we see that ultimately DNA or RNA, how it tells, let's say this is one strand of the DNA. Okay, then this portion, very small portion, it is known as a gene. And on the gene, there are located some specific codes. Ultimately, these codes are also made from nucleotides such as A, T, C, and G. That is adenosine, guanine, cytosine, and thymine. In the RNA, it is AUCG, uracil. Okay, so why I am telling you this all? Because it's very important for us to know. Earlier question has come from the CRISPR-9 exam or CRISPR-9 enzyme or CAS9 also. So let us see. So this is the basic structure of the DNA. Basic structure of DNA. So you can see the ladder here or the backbone. It is made up of sugar and phosphate. Sugar does not mean the sweet sugar we eat. Sugars are kind of compounds, specific compounds. Alright, they have various different chemical properties. Then there are base pairs attached with the sugar phosphate. And to base pair, there are ATCG or AUCG, they are attached to each other by hydrogen bonding. Alright, let's move forward now. When you change any of these genes by something, they become genetically modified. So what will GM do? If you, let's say, insert one more gene here, this is the DNA, let's say. So if you insert a gene here from some bacteria that makes a protein, that makes, let's say, BT protein, Bacillus thuringiensis protein, then it will go to the stomach of this parasite, whoever is eating this crop, and burst its internal walls. So this is all about the GM process. Let's now move forward and see the mustard. What is mustard? Alright, this year also a question came about the MSP offered on mustard. So let's see about the mustard. So it's a significant oilseed crop. Alright, this is the first and foremost important portion about this. Cultivated in various parts of India, primarily during the Rabi season. So this is a Rabi crop, alright, winter months. That means it is sown in the winter months. Alright, and harvested in the summertime. So the major states that produce mustard in India are Rajasthan, Uttar Pradesh, Madhya Pradesh, and Gujarat. There are two types of mustard crops grown in India. One is rye, that is Brassica juncea, and Sarson Brassica nubus. Mustard is primarily grown for its oil-rich seeds, which are used to produce mustard oil. Mustard oil is commonly used for cooking in various parts of the country. The climatic conditions are that it thrives in temperate climates. It requires cool temperature for its growth and development. Oftentimes, you will see that the crops that grow in the cool temperature, they have warmer properties on body. All right. And those which grow in the summer months, they will have little colder properties on the body. All right. So soil requirements. So well-drained and fertile soils are ideal for mustard cultivation. Sandy, loam and loamy soils are preferred. So this is very important, that which type of soil is required for the cultivation of mustard. It's usually sown from October to November and harvesting takes place between February to March. Now let's see about MSP. So government has increased the MSP of Rabi crops for marketing season this year to ensure remunerative price to the growers for their produce. The absolute highest increase in MSP has been approved from lentil at 500 per quintal, followed by rapeseed and mustard. Okay, so government does offer MSP for this and it's rather high, 400 rupees per quintal. Okay, a quintal means 100 kg. All right. One thing you remember, a ton means 1000 kg. Please remember the units that are used in the Indian agriculture system, especially hectare, that means 100 meter by 100 meter area. Okay. Okay, let's come to the GE mustard now. So genetically edited mustard, it's not modified. So oilseed yield not only for oil for cooking and frying, their so-called meal, the residual cake after extraction of oil from the seeds, is a protein source for the livestock, poultry and aqua feed. So this is very important. You must have seen the mustard bread cakes types. Okay. What is yellow cake? Please tell me. Yellow cake is not related to mustard. Please remember, it's related to nuclear science. So tell me what is yellow cake? So India most significant domestically grown oilseed is rapeseed, that is mustard. Its share in the country's production of vegetable oils has been estimated at 42.6%. It's more than soybean and that of meal at 30.3%. As per the US Department of Agriculture's data for marketing year ending September 2023. Mustard seeds have high level of glucosinolates. This is the one issue. A group of sulfur and nitrogen containing compounds contributing the characteristic pungency of their oil and meal. While that limits the oil acceptability among consumers, especially those preferring cooking medium having less strong flavor and odor, the problem is even more with the meal. Rapeseed meal is unpalatable to the poultry and pigs while having to be mixed with fodder grass and water for giving to cattle and buffaloes. So this is one issue because of this sulfur and nitrogen containing compound. What is the name? Glucosinolates. It become unpalatable to pigs and poultry, and for other animals it has to be mixed with water and fodder. Okay, so besides reducing their feed intake, high glucosinolates are also known to cause goiter and internal organ abnormalities in livestock. So this is one problem that we need to eliminate when it comes to the mustard. So a lot of efforts in the past 2 decades, including by scientists at Delhi University, CGMCP, and the Indian Council of Agriculture Research has gone into the breeding of rapeseed mustard lines, so-called canola quality. So what is this canola quality mustard? So question can directly come as this, that what does canola quality mustard offer? Okay, canola quality, let's say it will say that it saves water during the production. It has reduced glucosinolates. The, let's say, production is much, or whatever, there's a fourth option also. Only 1 and 2 are correct, only 2 are correct. Okay, so answer is that glucosinolates are reduced. So the dry seeds from the normal mustard cultivated in India contain 120 to 120 parts per million of glucosinolates. This is against the sub 30 ppm levels in canola seeds. By lowering the glucosinolates content to the same dry seed weight concentration, the scientists have bred mustard lines whose oil and meal match the standard of canola quality rapeseed in terms of pungency. What is pungency? Pungency is a smell, okay, because of the sulfur. It is sharp to the nose. So when you cook something in mustard, it is little sharp to your nose. Alright. Large scale cultivation of these canola low quality glucosinolate mustard lines hasn't taken place yet, as a major reason being their vulnerability to pest and diseases. The same glucosinolate that limit the palatability of the meal and exports true protein potential are also key arsenal of basic of family crops. Okay, so what happens that this cabbage, cauliflower, broccoli, they are all modified mustard only. They are modified from mustard plants by selective breeding. This is also one important fact. Furthermore, this pungency acts as a repellent to many pests and diseases. Okay. So cabbage, cauliflower, broccoli, they have all the same tool against invading pest, pathogen and termites. So that's where the role of novel breeding research comes in. So glucosinolates are synthesized in the leaves and pod walls of mustard plants. The translocation and accumulation in the seed happens through the action of glucosinolate transformation or GTR genes. So what we need both of the things. All right. What we don't need to eliminate the glucosinolates completely. We need them in leaves where they protect us from diseases, but we want to stop their transfer in the seed. Okay. I hope you all know what is seed. So it's a part of the flower. After the flower is shed, there is a seed and a fruit. So we don't want this to transfer into the seed. So we must stop its translocation. And this is controlled by a GTR gene on the DNA of the mustard plant. Let's say this is the GTR gene. Okay, so what we need to do, that there are 12 such genes under two distinct classes of GTR1 and GTR2 with 6 copies of each. So the scientist named Bisht and the fellow researchers have done is to edit 10 out of the 12 genes in Varuna. Varuna is this, please remember this important term. Varuna is a high yielding mustard variety. For this, they used CRISPR 9Cas9a gene editing tool, deploying an enzyme which acts as a molecular scissor to cut the DNA at precise targeted locations of the gene. So DNA is being cut and furthermore it is being edited. The quality of the line of the mustard being used is Varuna. And then letting the natural DNA repair process to take over. So which enzyme was used here? CRISPR 9 or CS9 editing to create mutations and change the nucleotide or basic building blocks of sequence of DNA in the 10 target GTR genes. By doing this, their encoded protein responsible for transport of the glucosinolates to the seeds were rendered non-functional. So now what we have achieved, that in the leaf there is still glucosinolates keeping away the pest and the termites. Okay, but the seed, the flower or the seed is not having the gene because not having the glucosinolates because we edited the gene from the different quality, Varuna quality, and now the protein, that is transport protein, it is being stopped, it is not being produced anymore, or it is non-functional. Okay, there are 3 types of expression of any gene: protein, functional protein, non-functional protein, and non-protein. This is often used, non-functional protein. Okay, protein which is produced but is non-functional. So the seeds of the resulted targeted gene-edited Varuna mustard quality as glucosinolate content well below the 30 ppm canola quality threshold. So we have achieved this. At the same time, the other plant parts, especially the leaves and pod walls, have significantly higher glucosinolate accumulation because now the transport is not being achieved. So we achieved both qualities in very good manner, that there is enough of the glucosinolate in leaves, keeping the disease away and pest away, and seed does not have this. Okay, so both probo. Alright. So the GTR edited low seed, high leaf glucosinolate line were tested for resistance against the virulent fungal pathogen Sclerotinia and the insect plant Spedoptera linora. These edited lines displayed defense response against this at par with or better than that of wild type mustard. The higher glucosinolate concentration in the leaf and pod walls during their impaired transport to the seeds clearly had a role in this. Okay, so more pungent smell and more, let's say, resistance against the pests. Okay, the new GTR genes edited mustard lines are transgene free or non-genetically modified. They contain no foreign genes, the Bacillus thuringiensis bacteria in cotton. So basically it's not GM, it's GE. That means no new DNA is added. No new DNA is added. Okay. In the GM hybrid mustard, DMH. So this was a very recent mustard that was come up with DMH 11, Haritadhara 11. So this was GM also, but not was approved by the court. A low seed, high leaf glucosinol mustard line developed by Bisht and the scientist genome edited, or GE, as opposed to GM. Okay, while the CS9 enzyme derived from the Streptococcus pyogenes bacteria was used to cut the DNA, the protein is segregated out in the subsequent generation. The final GE lines contain no CS protein and are transgene free. This is very important. Okay. GM crops are currently subjected to stringent environment release, not just for commercial cultivation, but even field trials and seed production. Such release is contingent upon clearance from a special GAC, under the Ministry of Environment, Forest and Climate Change. The GAC green signal is itself not binding on the union government, which gives the final nod. Okay, so this is the issue with the GM crops. That is also one reason we have come with GE crops, and they are far better in the results. Now let's see why is this work important. So India is a huge importer of edible oils. Okay, this was one of the major reasons of inflation in the last year. These imports were valued at 20 billion, okay, or almost 21 billion during the fiscal year ended March, which making over 60% of the country's consumption requirement. Given the massive foreign exchange outgo on account of imports, there is a dire need to boost domestic oilseed production through focused breeding for improving crop yields, pest and disease resistance, and production quality. Mustard and soybean are India's most widely cultivated oilseed crops, planted annually on 9 million and 12.5 million hectares area. Again, hectares has come into use. So please remember all these units. Its higher average oil extractable content, 38% versus 18% for soybean, makes mustard the bigger oilseed crop. Okay. While a source of both fat for humans and protein for animals. GM hybrid mustard and the new GE low seed high leaf glucosinolate lines are major plant breeding advancements from Indian scientists that can go in some way toward bringing down the dependence on imported vegetable oil. So we can produce more and use it more. Okay, the one thing that we need to produce something more is having the demand for it. So more demand will rise when there is low glucosinolate in this, and it is more edible to the poultry and pigs. So we'll be able to produce more also. All right. So this was today's lecture. It was a short one. Next one, we'll address some more issues. So keep studying and keep growing. Thank you very much for your time. Have a good day.